All right. So first of all, recapping the sarcomere. All right. Sarcomere is this single unit, right, inside of every myofibril, right? We have many repeating units called the sarcomere. Let's go click back actually for a second.
Wait, the sarcomere is one section. How long is it?
So if we look here, so like this is the whole myofibril, right, in here. So it's divided into these small sections called sarcomeres.
Oh, so basically light, dark, light.
So within the sarcomere, you have light and dark both, right?
Yeah,
So
patterns like that.
yeah, this pattern, right? So each one of these basically like there's sarcomere, every single one is a sarcomere, right? Blah 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 blah. Okay, so if you're labeling, first if you label it in terms of the coloration, you would say that the light band goes across from half this sarcomere and half of this sarcomere, right? So it's where the myosin ends and the myosin begins, and it includes the Z line or the Z disc. Whereas the dark band is where they overlap, where myosin and actin overlap one another. Okay, so that's if you're just looking for coloration. Now this, it's a good question from the other classes. This is not really, it doesn't mean anything. It's like just what you would see, right? So it's just based on seeing it under the microscope. It looks dark because of overlapping. It looks light where it doesn't overlap. So it's very clear in this microscope image below, right? The light part extends at the edges. While we're talking, I'm going to start by drawing some things. While I'm talking, I'm going to start preparing my drawing. Okay?
Hello, are you here? Okay.
Hi.
He's alone?
Yes.
Okay, great. I'm just letting you know, CDC is here.
Okay.
So if anything, he's safe, but just in case the other science lessons are full in the labs, if you can pass the word around. Thank you.
Okay. Disease control.
Oh.
Okay, so that's in terms of the light, but if we want to talk about the actual material, so we should say that first, the borders, right? The two outside borders are the Z disc. Okay, so you have Z disc on the end, Z disc on the end here.
Wait, so the vertical one is Z.
Correct.
And actin is the...
Actin and myosin go horizontally.
Oh.
Yeah. So Z disc, that's where the actin is actually attached to it.
Oh, there's two Z discs.
Yeah, they're the outside edges.
So this and this are both Z.
Yeah, that one and the other side.
Wait,
This
this and this?
and yeah.
Oh, this and this. What's this? This is just a line.
Squiggly thing?
No, this is...
I mean, this is the,
Oh.
this is the disc.
Oh, okay.
So the Z disc is the outside edge, but obviously like it's shared, right? One Z disc shares with this side, shares with this side, because they're both attached to it. The actin is attached to the Z disc, and then you also have in the middle some proteins that help for anchoring called the M line, okay, or the M disc. This one here. It's a little bit less seen. So Z line or Z disc to Z disc is one sarcomere. And actin attaches to that. M disc or M line is down the middle. That's easy to remember. M line, M disc, middle. Then I
Oh,
band.
and light band.
I band. I band is just another name for the light band. So here you can remember because I is in light. Okay, and in that the same thing. It's only actin, right? So it involves the Z disc, and it's just where actin starts before it meets myosin.
Actin and Z disc.
Correct. And then A band is the dark. So again, you can remember it from A and dark. This has overlapping actin and myosin. And then another name is the H band. The H band is is the myosin filament only. So even within, it's not shown here. Let me see. It's not shown there either. Here. H band is within the edge of actin to the edge of actin.
Yes.
So where only myosin is found. Okay. So if you went from middle to outside, you would say, like it goes Heiz.
Sounds like Chinese.
Heiz. Right? M is your M disc. Then H is between myosin to myosin. A band is from actin and myosin together. I band is on the outside, where this doesn't go, and then it's bordered by the Z disc. How are you doing? Ventilation's good. Windows are open. Are they open? They better be open. Otherwise we'll get in trouble. All right. Okay. Sarcomere wise, let me see.
This one, this.
Yes.
This is and this.
So go from the center. So M, then H. Yep. Good. H. Yep. Okay. And then
This is A.
A. Yep. Good.
This is I-bound.
From there to the like the next one. Okay, we would go across, right? Because then the next one would start there. Yeah. And then myosin would only be like here, essentially. Yes, exactly. So that's the light part, I band. And then Z. Oh yeah, Z. That's another Z. Good. Okay. So now this is the most fun part, I think. The next part is, how does this thing, this sarcomere, how is it involved in the contraction of muscle? Because what we're going to see is this little thing, this little block, okay, is going to contract using something called, we already did that, sliding filament theory. Okay, so sliding filament theory says that this sarcomere, right, one Z disc to one Z disc, will get shorter.
So it's fear.
Okay, this, I mean, it's the best we have so far, just like any other theory. It will get shorter. Okay, the filaments do not change in size. The myosin stays the same length. Look, if you follow here, this is still the same size, right? Actin has not changed in length from either side, but it has gotten closer to each other, right? This is the sliding filament theory. It's the actin is actually going to be pulled towards the center. Both sides are going to get pulled towards the middle.
So it's the Z-band that's moving.
In a sense, yes. The Z discs are getting closer to one another. But the weird thing is,
But it doesn't like break or.
exactly, all of them are doing the same thing, right? So what's happening is that collective getting shorter is the contraction of one myofibril, and if all the myofibrils are doing the same thing, that's the contraction of the entire muscle. Right? The whole entire structure becomes shorter. So as that happens, they become completely overlapping, right? So we can see here that the A band is almost the whole thing is A band, and very little I band anymore, right? The light band is gone. Here the I band is much larger. There's no light band anymore in the center. Okay? So this theory is what we're going to explain today. How does the myosin and actin interact with each other to make the two sides of actin come towards the middle? Oh, I forgot my notes. What did I do with my notes? Give me 1 second. Leave that there. It's so those obviously they're not just floating, right? Like the protein's not just floating in midair, so it has to be connected to the.
Sprinkling.
It's like a protein that helps with attach, because like otherwise this would just be like in midair.
Oh.
So it's also connected to the Z disc, but more
Yeah,
light, more lightly. No.
Oh.
Okay, ready? This is where it gets fun. The muscle shortens. They don't change length. They just slide past each other. Okay, so it looks in context a little bit like this. I'm gonna draw it later, but I wonder if I'll just have to keep checking if Janaki is here or not. Oh, that's not what I wanted to do. This is what I wanted. So yeah, you can see here, so there's my M disc, right? This is the contracted version, this is the relaxed version.
That's the top image.
This is the this is the contracted. This is matching with each other. So you can see here you have an H band, right? And there's the A band is wide, and here the I band is big. Here the I band has become smaller, right, compared to this.
Oh.
So the
There's
whole-
no reach band.
No more H band here. Okay, so all of this happens because of the interaction between the actin and myosin. Okay, actin and myosin are going to perform this like very, I don't want to say complicated, but very intricate dance, okay, with each other to make this sliding filament theory work. The way that I try to sort of illustrate it is this. Imagine that the myosin, I'm going to kind of stand here so that people who, if you watch the recording, the myosin has these heads, right? These like little pieces that stick out. And that head is what's going to interact with the actin. And you imagine that like the myosin, okay, is a long filament, but there's parts that stick out. What it's doing is it's essentially grabbing the actin and then pulling it towards the middle. And after it's done that, it lets go, reaches out again, grabs it, pulls it. Reach, grab, pull. Like this, and keeps pulling so that both sides are coming closer to the middle. And it's happening below too, right? Reach out, grab. Pull, 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 pull like this.
There's no one pull, multiple.
There's going to be multiple because if you look back to what the actual myosin looks like, there's so many heads, right?
If it pulls, doesn't the myosin get shorter? But you said it doesn't get shorter.
Myosin doesn't get shorter, it just does this. Are my arms getting shorter?
This is getting shorter.
No, it's not. It's like, I gave the example, it's like when people,
This is like a lever that pulls out like this.
it's like the correct term is like a ratchet. I don't know if you know what a ratchet is. Like something you can tighten, where you tighten it and then you put it back.
Oh.
Tighten and put it back. Right? Like,
So it's a multiple contraction.
yeah. Or
One muscle contraction does this.
many, yeah, to the middle.
That was just one.
No. Okay, we'll get to that as well. So the other way of thinking of it is, you know, when people are, the image that comes to my mind is like fishermen, where they go out and they put the net in the water and then they like bring it back to shore, and then there's a bunch of people pulling it,
Yeah.
right? So when I reach out and grab, and then I pull, the next time I pull, I don't move. Only my hands go forward and grab the next rope, right? And pull. But either way, it's coming towards me, right? I'm not like dragging this back and moving. I'm just pulling, then reset. Pull, reset. Pull, reset. That thing's still coming towards me. So that's what each head is like fixed in place, and it just goes like that. Okay? And this would happen along every part of this. So this would grab here. This side is grabbing this side. So they're pulling both ends.
But then the question is, if there's definitely not one
Head.
there's they're both pulling at the same time. The other side is also pulling the opposite.
Yes.
So what would-
So it's not going to break apart, but the collective is that the whole entire fiber compresses upon each other. right? These two are coming together, but also these two are coming together. The
So it's
collective,
like this.
yeah. And then the next one and the next one. So the whole entire thing.
So they don't stay in place. They move. Whatever.
I guess the Z disc kind of, not that it moves, but it can be brought closer.
Myosin moves, right?
Only the head moves for myosin.
But if it doesn't, the whole thing, if it doesn't move, then how does the whole thing like end up contracting?
Oh, I suppose as a unit, the entire thing, yes. I thought you meant myosin on its own. Myosin on its own won't go anywhere, right? Because it's fixed in place.
Oh, I thought it was just ripped apart.
No. Because that's why you asked about those proteins holding it in place, right? So it's not going anywhere. It's just that like the actin that's attached to each Z disc, those things get closer to one another. Okay, so here's what we're gonna do. I'm gonna go through and illustrate for you how this works. It takes a little bit of time for me to draw everything, but let's try. Okay, first things first. I'm gonna draw, now we're gonna use the other handout, okay? The other handout is the diagrams of, that one. And I tried, when I did it with the other group, I started with the third one. It's just the way that, I don't know, to me it was more convenient. And on the back there's some like terms that you can use to help fill it out, or you can just do it as I'm writing. Okay. Maybe also what I should, let me not make the same mistake. Last time I only showed the video at the end. This time I'll show you the animations first. Okay, now the order that I want to show them is this one first. Okay, ready?
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the
I should have said, this is where we can connect neurons unit, nerves unit with muscles unit. So you can see there's the signal, action potential is coming. Then you have the release of calcium from the sarcoplasmic reticulum. This is a combination of the two units.
T-tubule. The action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
Okay, a lot of terms. I know.
Give me a sec. What's a...
Don't worry. I'm going to explain everything as well. I just want you to see this in motion, because it's hard for me to draw the motion. So watch first.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
But this motion doesn't use ATP.
You saw ATP.
But it's when it's come back.
Correct. So it's a little bit counterintuitive. It's like when it reaches out, it needs ATP. I'll explain that as well.
The original position is this.
Yes.
But then if you relax, doesn't it?
I know, but it's not your muscle that we're talking about. It's just that myosin filament, right?
So technically,
In a relaxed
Right.
muscle,
Like,
They're like this.
and then if I do this, then it's like this.
Yeah.
What? What?
actin and myosin myofilaments lie side
So
by
watch
side,
this.
and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
Okay. So now let's try to illustrate. Okay, welcome. Are you there? Hey. Okay. All right, so this might be a little bit crazy. What you're about to learn. So follow along as much as you can and then go back. I'll upload the video today. Sound good? Okay. All right. We are, do you have, if you check no, I'll send it again. I'll go to WeChat. Okay, this will be helpful. I'm sure of it. This is a good idea.
I like how they give me this
Yeah.
today, but nobody uses it.
Okay.
I'm going to use it for myself.
All right. I am WeChatting you a PDF also. You can use it to follow along. When you open it, you'll see 4 pictures or 4 like images, like a cartoon. The way I'm going to start it, I'm going to start with the 3rd image. Okay? Okay. All right, here we go.
Oh, that's why it looks weird.
And if you're looking on the PPT, it's slide number 37.
That's why on the diagram it looks like it's fall apart.
Yeah. Let me switch this to here now. Okay. So here's the deal. So we're talking about the interaction between myosin and actin. Okay? Shoot. How do I want to do this?
There's it.
Okay. There's ATP. There's blah, blah, blah. Where does calcium come in? It doesn't show it there. All right. So here's the deal. I'm going to draw myosin like this. Okay. I'm going to draw the head like that. Okay. And of course, there'd be more of these like this. And there'd be some going down. Okay. This is the whole thing. This is my myosin. Okay. These are the myosin heads. Okay, the black thing is, the black thing is actin. Then we have on top of this, green is tropomyosin. And the red is troponin. Okay, so for context, the way that I'm starting this drawing is is the 3rd one, yes, in a sense, somewhere between the 2nd and 3rd one. Okay. So what we have here is I have the myosin head already connected to the actin. Okay. Now in this situation, this is like post post-muscle contraction in a sense. This myosin attached to the actin is called a cross bridge. Okay, so let's say this. This myosin attached to actin, this is called a cross bridge.
Attaches to the actin or the.
It's attached to the actin. Yes.
protein?
No, it's attached to the actin.
Ac-
So
okay.
let's say this. I'm going to put point 1 here. ATP is needed to break cross bridges. Okay? ATP is needed to break cross bridges. Meaning that this myosin head cannot detach from the actin unless ATP shows up and binds. Okay? So in this situation, then I'm going to simplify my actin. Okay, I'm just going to do this. Okay, there's my actin now. And I'm just going to show one myosin head here. Okay, and we have ATP attached here like this. Okay, so there my cross bridge has been broken. Okay, and I can re-add all my stuff here. Okay, so there's a space now between the myosin and the actin. But in order to like grab it and pull it, it needs to grab on again. Okay? So the myosin heads are detached. Then, in order for this to reach out, we need ATP to hydrolyze. Okay? So that means step 2 is ATP to ADP plus PI.
That's a fourth image, right?
Yes. Sort of fourth leading into the first kind of thing. Okay? So when ADP hydrolyzes to PI, what's going to happen is then the myosin head becomes, the term that we use is it becomes cocked, meaning it like reaches out or it like flexes. Okay? And I know this doesn't look like flexes, but this is kind of like flexing. Okay? So when this is broken apart,
My flex.
the myosin head is, and this is the word that we're going to use, is cocked. Okay? So what does that look like? It looks like, I guess I'm writing this beside the wrong thing. I'm going to go this way. Okay? So it looks like this. Okay? So it like moves like this out to the side, and we can say that there's now two parts, ADP and PI. Okay? So it becomes cocked. Now at this point, I'm intentionally showing that the myosin head is not yet attached to the actin. I'm doing that on purpose. Because there's another thing that is required. At this point, I stopped drawing them a little bit, but in blue up here, I was drawing a little patch, okay? Like this. Okay? What that blue is representing is something called a binding site.
So this doesn't pass on doesn't bind on that.
No, there's specific locations on the actin, like regular locations, where myosin can bind to the actin. Now, in normal context,
So a hook and something.
sure, you can think of it like that. Yeah, like the ring that the hook can grab onto. Sure. So normally, let's call this point 3. This is gonna get really convoluted, but okay, we'll see if you can follow along. Let's make sure the camera is following along as well. Okay. So, in normal situations, the tropomyosin covers the binding sites on actin. Okay, so this green filamentous tropomyosin is usually laid on top and covers the binding site so that myosin cannot bind to actin. The only way that we can uncover the binding site is by moving the tropomyosin aside. The only way to do that is when calcium is released, calcium binds to troponin and pulls tropomyosin out of the way. Okay? So we can say calcium binds to troponin, which
It pulls what away? Myotropin?
tropomyosin, which moves tropomyosin, therefore exposing binding sites. Okay, so how does that look? Again, I hope that this is visual, can be seen. Okay, so what does this look like? We would say that, here's my actin again, a double filament actin. So now calcium shows up and pulls the tropomyosin away because the calcium binds onto these troponin globular proteins. All right, calcium, calcium, calcium. And what does that do? Now, the binding site is open. And what does that do? If the binding site's open, if the myosin head is cocked, it can form a cross bridge. Okay? So then the myosin head is able to attach.
Still the ADP, right?
It's still, yeah, we've kind of taken a little, we've frozen time for a minute here. Okay? So it can now form cross bridge. Myosin binds to form cross bridge. Right? It's still, we can say that still ADP and PI are attached here. Okay, so this is like a if there's ATP, we can break the cross bridge and cock the myosin head. If there's calcium, also the binding sites will expose and you can attach the myosin to the actin.
Where do
But-
you get the calcium?
The calcium is stored where?
Is it in the nerve thing? No.
No, this nerve signal will be sent. There's a link between the two, yes. But what part of the muscle fiber stores calcium? One of the new parts of the muscle fiber we learned. But it sounds like something from a regular cell.
Something particular.
Yes.
The sarcoplasmic reticulum.
Yes. The sarcoplasmic reticulum has calcium inside.
Okay.
Okay?
The white, the yellow thing.
Right. Okay, so now, again, sites open, so now this myosin head can grab the actin. It's connected. But in order for the shortening, right, for the actual contraction, it needs to now come back to the middle. Yes? So, how does that happen? So the next step, let's go like this, I guess. And I'll check again to make sure you can see. Yeah. Okay. Move these out of the way. Okay. So the last stage is the ADP and PI will release. Okay? If the ADP and PI release, you get the swivel, or kind of like the myosin head returns back to its like original position, right? So the swivel means going from out here towards the middle.
This is original.
Yeah, I guess so. Or like towards the center. It's more of the relaxed. Remember we said like this is relaxed and this is flexed. It's like opposite. So we can say for release of ADP PI causes myosin head to swivel. Okay. Pull towards the middle. The center, let's say, center of the sarcomere. Right? Pulls towards the center of the sarcomere. So how would that look? Myosin head is now back to its original, like this. Right?
It's still bonded, right?
And it's still bound, exactly. Yes, that's very important. Okay, it's still cross-bridged. Tropomyosin, everything is still there. And then everything from here on out just depends on the availability of materials, right? So is there more calcium? Is there more ATP? If so, and is there a signal? Then it will continue to do this. So then we say ADP and PI get kicked out. And this is returned. Okay? Cross bridge remains, right? Unless there's more calcium or and more ATP.
The calcium is required. Break it?
No, but once it's broken, if there's
You need
no
the calcium to bind it.
to open the
To-
site,
yeah.
expose the site. Yeah. So if it breaks it, let's say you have ATP but no calcium, it doesn't matter, right? Because ATP will release the cross bridge, and it'll go like this, and it'll be like, hello, what do I grab onto? There's nothing to grab onto. If there's calcium, then you can open the binding site, but it doesn't matter because this will just be stuck as a cross bridge. Okay? That's it. That's it? That's it.
Yeah.
Sorry, Janaki. I should have brought you closer. I don't know if you can see that drawing well enough. It will be on the video a little bit better, I think.
Yeah, so actually, it's kind of
Yeah?
blue and but it's still seeable, yeah.
Drop it down a touch. I can't see the video of myself, so I don't know what it looks like. No, that's not what I want. Oh, there. So this would be better like that. Okay. Right, so again, if you look at the slide, it says here that this contraction, slide 37, okay, it says that, it says here, this happens up to 5 times per second.
What?
Right? Yes. This whole cycle happens up